Okay, thanks, Brian. I'm a neurobiologist, so it's a real privilege for me to be here at this meeting, also to be a member of the CFAR network, because it gives us a chance to step back from what we do in the lab and think about the big picture questions. What I'll try to do today is give an overview of mechanisms in the brain that seem to control developmental windows of plasticity, what we call critical periods. But of course, the motivation for this understanding is something that you're all familiar with. We've known about it since the ancient Greeks. And something about early life experience is special. It shapes our brain to make us unique individuals, and it would be exceedingly important to try and understand this process, if only for the fact that as a parent in one of these weird societies, these early life experiences seem to be very important. And we want to know, how does this come about? How critical are these windows? And can we actually understand them well enough to perhaps manipulate them and make use of that knowledge in cases where we need recovery of brain function? So just to give you a flavor of what I'm talking about, probably many of you speak multiple languages, not quite as much as Jared Diamond, perhaps, but this facility with language seems to be greatest early in life and gets harder with age. And so if you peer into the brain of someone who's bilingual using fMRI techniques, as Joy Hirsch had done already a couple of years ago, you see some interesting differences. So this is the brain of an individual who acquired a second language after the age of 11. And in particular, language-related parts of the brain, you see that the first and second language activate discrete spots when processed. And this is different from people who are raised in a multilingual environment who seem to have shared resources for processing essentially two native languages. And this study was quite interesting because they looked across a number of language pairs, grammatically close ones as well as distant ones, and seemed to come up with this same milestone across subjects, namely that something about exposure before the age of 11 was instrumental in producing this difference in the way in which the brain is used. And if you step back from that for a moment and consider when second language learning is typically introduced in our societies, it's quite interesting that it's relatively late, and sadly in our country here is especially late, suggesting that we've sort of missed this sort of critical period, perfectly timed for the end of this moment when a second language could be introduced. And this is already an old slide, and hopefully things are changing. But from a neurobiological point of view, understanding these milestones, just identifying them in humans, is a major step forward for policy-related issues. But it's extremely difficult, as you can imagine, to dig into the mechanisms of why circuitry is connected differently depending on the age at which these stimuli are provided. And so it's with this in mind that we turn to animal models. And again, you see a slide copied from Chuck Nelson, which highlights this notion of critical periods as a multi-step process. So it's not that there's one monolithic window of plasticity, but most likely all brain functions will show some nature of critical periodness, and these windows will come on at different time points, and that higher order functions probably emerge later than primary sensory functions because you need to set up those earlier functions, and in a cascading or hierarchical fashion you can build up higher order brain functions which may remain open in terms of plasticity longer than the first filters to the world. These are, of course, windows of opportunity if you think about learning language, for example, but they're called critical periods most acutely because deprivation studies have shown that they are, in fact, critical. So we consider these also windows of vulnerability, and there are disorders that seem to be enduring if particular stimuli are not provided at appropriate times. And in the case of animal models, where we can't rely on verbal feedback from the animals, we tend to find examples of this in primary sensory or motor areas where we can measure in the performance of the animal. So today I'll talk mostly about primary cortical areas that deal with vision or hearing, and the consequences of deprivation or altered sensory experience in these areas. So take the example of language. You'll hear a lot more about it in the next talk from Janet Worker. Babies are born into the world not knowing where they will land, but then are showered with sounds, native and non-native speech sounds, as well as non-speech sounds, and somehow they have to sort through all of that. Well, we can do something similar with rats or mice by exposing them to tones or music and watch what happens to their map of sound space. And so this is a one-slide overview of the process. We call it brain plasticity, and probably the layman on the street would define it something like this, that plasticity in the brain is the ability to alter its function in response to experience. If we want to take this into an experimental cellular molecular setting, we might translate these words into something like molecules adjust connectivity patterns through neural activity. So abilities are read out in our talk today as particular molecules. Brain function is the reflection of particular circuits that are wired up. And experience, broadly writ, is a pattern of neural activity. In fact, if you were asked, how does the brain work, as Steven Pinker was asked on the Colbert Report, maybe you've seen this, he was asked, how does the brain work in 5 words or less? And he actually answered that question. He said, brain cells fire in patterns, which is a beautiful answer, actually. But what this definition lacks is the plasticity component. So there are 2 other catchphrases in our field that typically come up. These patterns can, in fact, adjust connection strength between neurons. And you've already heard these experimental examples in Michael Meany's talk. So one is the notion that neurons that fire together will wire together. So synchronously active pre- and post-synaptic elements will eventually strengthen their connections, something like LTP, long-term potentiation. But on theoretical grounds, you might imagine that this is not enough. Eventually, you might saturate the entire brain and everything would be wired together. So clever people came up with other phrases like out of sync, lose a link. And so that's the long-term depression kind of aspect, that things that are not correlated should be weakened. Of course, this is grossly oversimplified, but these notions are themes that pervade the study of critical periods, because early life experience played out as neural spiking patterns will eventually engage these rules of plasticity. So let me give you an example. If you raise a mouse listening to a tone passively, then you can, in fact, rewire the neural circuitry responding to the sound. And this is done by electrophysiological mapping. So what you're looking at here is the surface of the auditory cortex of a mouse, and each of these colored dots represents the best frequency that activates neurons next to an electrode in that region. These gray and black lines are blood vessels on the surface of the brain as landmarks. And so you see that there's a nice rainbow spectrum of colors here representing the different kilohertz frequencies of tone played to the animal. If you raise a mouse listening to one tone over and over again, let's say 7 kilohertz, this lime green color, then you can expand that representation. So expand this small representation to a larger one at the expense of neighboring frequencies. And what's interesting is that this can happen just by passively playing the sound to the animal during a rather narrow critical period from postnatal day 12 to 15, and doing the same thing in an adult mouse does not change its auditory map. So this is what we would call a critical period phenomenon, and you can hear a lot more about the auditory critical period from Anne Takesian, who has a poster later this evening. I'll switch gears to another example. Classic example is the visual system, made famous by Hubel and Wiesel at Harvard Medical School 50 years ago now, where they started to explore where do the inputs from the left and right eye first converge in the brain. And this, they found, was in the primary visual cortex of cats and monkeys. And if you inject a tracer into one eye, they will label inputs in roughly even fashion in normally raised cats or monkeys. So what you see here is the primary visual cortex labeling one eye in white, and the negative inputs are the ocular dominance columns, as they're called, of the opposite eye. So in a normally raised animal, there's a roughly 1-to-1 relationship of the two eyes' inputs. But look at this. If they patch one eye during a critical period early in life, and this was to simulate what happens during discordant vision, such as a lazy eye, as many people have at birth. If they patch an eye during a critical period and then inject the open eye, you see a dramatic expansion of the territory occupied by the open eye at the expense of the deprived eye. And to me, this is the most fascinating aspect of a critical period, that simply covering an eye, altering sensory input, will physically rewire the brain permanently. And so after a critical period, it becomes difficult, if not impossible, to restore this connectivity pattern. And as a result, the functional vision through the deprived eye is decreased enduringly. That's called amblyopia. Using mice and various molecular techniques, we can now identify what are the steps that take you from a normally connected circuit to a grossly biased one, and it involves the pruning of spines through the secretion of proteolytic factors, for example, the cleavage of cell adhesion molecules, the motility of these spines to the point where they eventually get pruned away. Pruning is a very important process here. Then these factors fade away, and then there's a regrowth phase where the open eye inputs expand to attract new synapses. So this is a highly orchestrated developmental process. Most of these molecular events do not occur in the adult, so patching an eye in the adult will not produce these changes. And so the question for us in this talk is, what is timing this to an early window? And so I'll give you the three messages, mostly coming out of the study of mice. So amblyopia, as I said, is the enduring loss of visual acuity due to discordant vision, patching an eye. And the take-home message here is that there seems to be a dynamic change in excitatory and inhibitory components in the visual cortex, which actually triggers the window of plasticity. And then those pruning events can take place. And they would continue to take place, it seems, if not for another class of molecules, which we're calling molecular brakes, that come on later in life and clamp down and actually restrict or limit this plasticity. So I'll mention these, and along the way try to draw inferences for other systems. So let me explain to you how we do these studies. You can measure vision in a mouse by placing an electrode on the visual cortex and presenting black and white stripe pattern of ever finer frequency. And at some point, this beautiful visually evoked potential, analogous to the one you can measure in humans, disappears. And so you get an acuity threshold in these animals. If you then start to map when is the system sensitive to lazy eye or patching of one eye, you find that there is a critical period. If you patch the eye between postnatal day 20 and 40, which is hiding behind there, you get an enduring loss of acuity. So in fact, in these experiments, the eye is in fact reopened here, and the measurements are taken here, so the acuity is permanently reduced. But a similar deprivation in adult animals does not produce this amblyopia. So here's a window of plasticity that we can study and dissect using molecular genetic techniques. Mice are not particularly visual, but they are genetically tractable. So you can start to play with factors that you think are important. In the cortex of the brain, there are essentially two types of neuron: excitatory neurons and inhibitory neurons. The excitatory cells using glutamate, inhibitory GABA. And this is essentially the brain's version of the 80-20 rule. For decades, people have focused on these excitatory synapses made by excitatory cells onto each other. In fact, those are those spines that have come up in Brian's introductions and what I just mentioned to you, and the site of plastic change. But it turns out that a late developing inhibitory component, this sparse population of cells, is what turns on the sensitivity to this plasticity. And in fact, these are very diverse in their morphology and connectivity, so they have very colorful names. The ancient anatomists gave them names like chandelier cells because they look like the old candelabra chandeliers, where each of these little candlestick holders is in fact targeting very precisely the axon initial segment where action potentials, the spike generators of neurons, is situated. What I'll try to leave you with is the notion that only one particular type of cell may be involved in the visual cortex in triggering the critical period, and that's this basket-type interneuron making synapses on the cell bodies in a basket or nest-like shape. So what's the evidence to say that inhibition is so important? Well, I'll just put it all up on the slide. It turns out that if you delay the development of the GABAergic or inhibitory system, the sensitivity to patching an eye is delayed indefinitely into chronologically adult ages. And conversely, if you prematurely enhance inhibitory neuron development or function, you can accelerate the onset of this window of plasticity. So I'll give you some highlights. For example, you can knock out one of the two enzymes that produces GABA, the synthetic enzyme for GABA, and these mice fail to enter the critical period until you restore inhibition pharmacologically, for example, with benzodiazepines. So if you take a mutant mouse like this one, the critical period is waiting until they're given some Valium. Conversely, if you take a wild-type animal before its natural critical period and you give them Valium, they will turn on their plasticity window earlier than normal and then also shut it down earlier. There are environmental ways to do this. If you raise animals in total darkness and you never let them see anything, this also delays the critical period. And we now know that dark rearing delays the development of a key inhibitory cell type, those basket cells. An interesting recent study is this one here, published last year. Since inhibitory cell maturation seems to trigger the natural critical period, maybe you could do some kind of stem cell therapy. Could you take inhibitory cell precursors and inject those into the postnatal brain and recreate a second critical period? Well, that's exactly what they did here. They took embryonic transplants of the medial ganglionic eminence, where these interneurons are born, put them in the postnatal visual cortex, and found a second window of plasticity would arise only when this transplant reached a particular maturational age, about 1 month after their birth. And that maps beautifully onto the timing when interneurons normally develop in this system. So we finally have access to this critical period. We can move it to and fro. And what's interesting to me is that all of these different factors could be acting independently, but they seem to converge on one particular cell type. And here's a cartoon of that parvalbumin positive, PV positive, large basket cell, which contains all of those elements that were listed in the previous slide, and then some. And so this gives us a biomarker to look at different brain regions, for example. If this cell type is triggering one critical period in the visual cortex, maybe it's closely tied to other critical periods. And all you have to do is go into the literature, and lo and behold, you start to see that in primary sensory areas, I told you briefly at the beginning, the mouse auditory critical period starts around postnatal day 12. Well, that's ahead of the visual critical period I just mentioned to you, and the parvalbumin cells, sure enough, mature earlier in this system, earlier still in the somatosensory system that Brian talked about. And if you go into monkeys, which have higher order visual areas, the parvalbumin cells all along the neuraxis develop at later and later stages, consistent with these cascades, hierarchies of critical periods. Similarly, if you look at spine pruning across these different brain regions from the primary areas to higher order areas, you see that spine formation in these different zones occurs around the same time, but the pruning happens differentially and is much slower and less pronounced in the prefrontal areas of these monkeys, for example. So it could be we're onto something here, that this excitatory-inhibitory balance might be important for orchestrating not only the visual critical period, but hierarchical critical periods across the brain. And if that's true, maybe there would be some severe consequence if the excitatory-inhibitory balance failed to mature properly. And so it's interesting to note that neurodevelopmental disorders are increasingly being tied to excitatory-inhibitory circuit imbalance. Here's just one example where an epigenetic factor, the methylated DNA binding protein MECP2, which is linked, the causative agent of Rett syndrome in girls, has been linked specifically to the GABA circuitry when Huda Zogby's lab, instead of deleting the gene from the entire animal, deletes it only from GABAergic cells, is successful in recapitulating the disorder, including repetitive behaviors, epileptic seizures, and other features of autism in mice. If you look across a number of mouse models of autism, we find that consistently across both genetic as well as environmental models, they all have reported defects in parvalbumin circuitry, these basket cells. So we think that one implication is that the understanding of this excitatory-inhibitory balance in orchestrating normal windows of plasticity might play an important role in understanding these disorders. Now, in the home stretch, what happens if you've passed a critical period? Can you actually redo or undo this wiring in cases where you have a brain injury or, say, a neurodevelopmental disorder and would like to rewire the circuit properly? Well, this seems like science fiction. It's, in fact, the holy grail of our field. Can you understand this process well enough to reverse engineer it experimentally with obvious therapeutic implications? Well, recently, and just in the past few years, this has become possible. I'll give you a few examples. So one is, again, focusing on the very cell that I told you about. With age, as the critical period ends, they become enwrapped or encapsulated in extracellular factors called perineuronal nets. And these have been known for over a century to be extracellular matrix. It's like a saran wrap around the cell body and proximal neurites of these cells as the animal matures. And in fact, if you inject an enzyme that cleaves or disrupts these nets, it's possible to reopen a visual critical period, as shown by Maffei's group in this early paper. And more importantly, you can use that to restore vision to animals that were deliberately made amblyopic during the critical period. So the science fiction is starting to become reality slowly, and what's interesting is that they have to do something bad to the brain, remove the net, in order to improve plasticity. And so this gives rise to the idea of a molecular break. Maybe the brain wants to be stable after a certain point and goes to some trouble to make factors to suppress too much plasticity. Again, perineuronal nets, just like the parvalbumin cell itself, gives us a biomarker. So tomorrow you'll hear about birdsong and the beautiful critical period they go through in order to learn song. In fact, Teresa Nick's lab has looked and found, indeed, even there, parvalbumin cells in the key nuclei of the songbird system are acquiring perineuronal nets as their critical periods end. So again, we're hopeful that this is a generalizable principle. But to reopen plasticity this way requires an injection in the brain, something physically invasive, and seems a bit far off from therapy. And so I'll leave you with one other kind of break, which could be treated in a non-invasive way. And this is the notion of neuromodulation. So many of you have just come back from lunch and are perhaps dozing off, and your neuromodulatory systems are winding down. But when you're engaged and aroused, paying attention to something, there are chemicals that are released in the brain, cholinergic and aminergic systems in particular, that increase the signal-to-noise of those spiking patterns that Pinker talked about, and makes the salience of the input ever more important. We found that, in fact, there is a break on these modulatory systems. And there's one molecule that we produce in our adult post-critical period brain called LYNX-1, which has a structure that looks very similar to alpha-bungarotoxin, which is the active component of snake venom. So why in God's name would we make a toxin in our brain? In fact, Nat Heinz, who originally described this molecule, showed that it does bind to nicotinic acetylcholine receptors, just like bungarotoxin does on the muscle, and suppresses or dampens signaling through this system. We found that this molecule comes on late in life, and so it had all the hallmarks of being a brake. And in a number of studies, and this is perhaps the most clinically relevant, if you deliberately make an animal amblyopic by patching an eye and then reopen it, they don't normally recover, but mice lacking this molecule, they will recover. So you can spontaneously restore vision in such an animal. But what's interesting here is an opportunity, because we know that this molecule acts on cholinergic signaling. Maybe we can even rescue a non-mutant mouse, a wild-type animal, if we enhance cholinergic signaling during this time. And this seems to work by giving the animals cholinesterase inhibitors. So we're trying to find clever ways to translate our work from the mouse to human, and Janet will talk more about this, I think, in her talk. So I will stop here with the summary that the study of critical period, in particular the visual system, has led to this notion that there's a dynamic change in excitatory-inhibitory balance which turns on plasticity, and this would remain open if not for an extra coating of factors that consolidate the wiring. So the way we've shown that is to remove these breaks and allow plasticity in the adult. I should add that being part of the CFAR network has focused our attention on epigenetic control now, and in fact all of these aspects are very sensitive to epigenetic modification. So we have a lot of interesting opportunities ahead. So I think I'll stop there and just remind you to go to Anne's poster this afternoon.